So we just had a great week convening innovators across the genomics industry at the Illumina Genomics Forum. And during our Investor Day event today, multiple speakers will touch on how much our customers appreciated it and the groundbreaking technology that we launched at the event. Thank you for joining us here today, both everyone here in the room and everyone joining us virtually on the webcast. My name is Sally Schwartz, and I'm Illumina's Vice President of Investor Relations. Prior to joining Illumina, I spent the past almost dozen years leading investor relations teams at various companies. And before that, I held a variety of corporate and financial advisory roles. Before we get started, I need to provide a few cautionary notes. So on this slide behind me are really important disclaimers for our business. They include a statement reflecting the current hold separate requirement related to our Grail business. We encourage you to review them, and they'll be included on the presentation on our website. We have a really full agenda for you today. We're going to focus our conversation on core Illumina, and we have so much to share with you. We'll start the day with Francis D'Souza discussing Illumina's vision and growth strategy. Alex will then cover our innovation roadmap, including the product announcements that we made at our Illumina Genomics Forum last week. Bill's going to discuss where we are and where we're going in clinical markets, and then we're going to take a short break. After the break, Susan's going to provide a commercial update and let you know what we're hearing from our customers. And then Joydeep will wrap up the presentations with our long-term financial outlook. After that, we'll move to Q&A with all of our speakers. For those joining us via webcast, you're welcome to submit your questions at any time during the presentations. And so, we understand that you are curious about how our third quarter went. We just finished the quarter yesterday, and therefore we won't be commenting on it today or our full year 22 guidance. We'll have a full update for you in just a few weeks during our third quarter earnings call, which will be on Thursday, November 3rd. Before we move to Francis's presentation, I wanted to highlight a construct that's really central to the way that we operate. Environmental and social responsibility is core to Illumina's business, and today you're going to hear components of it across all of our presenter sessions. We understand that with our leadership position comes added accountability. Our 5 corporate and social responsibility pillars reflect our unique position to deepen our impact on human health by focusing on enabling genomics for good. We've been honored to have our work recognized by a number of organizations, and we're going to continue pushing forward with all these critical efforts. Similar to last year, we're going to be hosting an ESG investor event later this month to highlight our latest progress in each of these areas. We're going to have additional details for you on that soon, and we really hope that you'll join us there. On that note, let me welcome Francis D'Souza, Illumina CEO, to the stage for his presentation. Francis?
Hello, everyone. Thank you for joining us. As you saw in that video, we're coming off our sold-out Illumina Genomics Forum. It was a terrific customer conference where we launched a suite of incredible products in front of the in-person audience of over 1,000 people and an online audience of over 10,000 people. It's an exciting time to be at Illumina. Last week at the conference, I was speaking to the crowd, and we launched NovaSeq X, and I was challenging the audience to be bold in their ambitions. And I said, look, if for the non-Nobel Prize winners in the room, because there were some Nobel Prize winners, think big. Think of where your research could go. I don't know if you saw this morning the Nobel Prize for Medicine. It went to a researcher who did their seminal work partly on Illumina machines on the GA2. So didn't expect them to move that quickly. In this session, I'm going to talk about how genomics is changing human health and our strategy to continue to lead the large and high-growth next-generation sequencing market. Here are the key messages. Next-generation sequencing represents a $120 billion market opportunity, one that we're still in the very early stages of penetrating. Our strategy is to leverage our proprietary innovation to deliver NGS platforms and select applications to accelerate the penetration into this market and catalyze new, large, adjacent NGS markets. Our innovation leadership creates a powerful flywheel that represents a strong competitive advantage. We've attracted and built the deepest talent pool in the industry with a world-class leadership team. And this all sets us up well to deliver superior shareholder value through mid-teens revenue growth and expanding margins over the long term. So let's start where we are now and our position at Illumina at the center of the genomics landscape. Illumina's mission is to improve human health by unlocking the power of the genome. Over the last 2 decades, Illumina has emerged as the clear leader in research and clinical genomics, with an installed base of more than 21,000 sequencing systems across more than 9,100 customers in 150 countries. Our world-leading 1,000-plus person clinical genomics team has enabled us to get 72 product approvals from 58 regulatory agencies globally. Our terrific market access team has helped us get coverage for 1 billion lives globally for genomic testing. And we're working to increase that number to 2 billion by 2026. We have achieved this through our relentless focus on delivering breakthrough innovations. One of our values at Illumina is innovation is in our DNA. And as you saw last week, that's as true today as it's ever been. We have a portfolio of more than 8,400 patents worldwide, and we spent $885 million on R&D last year. More than 300,000 scientific publications used our breakthrough technology, significantly more than anyone else. And we're only just beginning. As we look ahead, I want to ground these conversations in the why, the immense potential of genomics that we've only just begun to unlock. Genomics will transform lifetime health management, improving health outcomes and lowering healthcare costs. We're working towards a world where newborns with suspected genetic diseases will have their genome sequenced as a frontline diagnostic. Carrier screening of parents will be a part of the reproductive process. Omic testing for screening and early detection of deadly diseases like cancer and neurodegenerative diseases will be routine. Our own immune system will be activated as the strongest therapeutic available. And pharmacogenomic testing will select the right treatment at the right level for patients. Therapy effectiveness and recurrence monitoring for cancer and many other diseases will be required at every progressive treatment line. Illumina's products will touch every step of this health management continuum. Each stage of this lifetime journey will involve genomic research, testing, and personalized drug development. Today, only 4 million people, that's 0.07% of the world's population, have ever had their genome sequenced, even once. We'll go from this to over 1 billion people being sequenced dozens of times over their lifetime. Several orders of magnitude more sequencing than is being done today. Now, to make this vision of lifetime health management a reality, we have to enable more discoveries, get more applications into the clinic, and make genomics a standard of care. Realizing this vision starts with research in biological discoveries, from identifying a new biomarker for diagnostic or therapies to targeted mechanisms for drug development. Each discovery must be translated into clinical utility. It must then become a standard of care. Since the Human Genome Project completed in 2003, we've seen clinical applications start to go from research through translation to standard of care. And as research continues to expand, the pace of applications continues to accelerate. Our platforms initially powered genomic research, which was our first addressable market. In 2005, our TAM included genomic research in areas like genetic disease and genotyping. By 2014, applications such as non-invasive prenatal testing, research disease diagnosis, and therapy selection for oncology emerged from research and were moving into the clinic. This increased our addressable market to $20 billion. Looking forward to 2027, we see another six-fold increase to an addressable market of $120 billion, driven by, first, the emergence of large new clinical applications, like cancer screening and minimal residual disease monitoring in oncology. The transition from single-point use cases in clinical care to multiple touchpoints throughout a person's lifetime for screening and disease recurrence monitoring. And the expansion of sequencing-intensive modalities, like spatial and single-cell. Most of this addressable market is in clinical segments. And today, these applications are less than 10% penetrated. We're focused on accelerating this penetration and estimate it will grow to about 15% over the next 5 years. Later today, you'll hear from Phil Febo, our Chief Medical Officer, who will review each of these clinical markets in detail. In research and applied markets, the current penetration is higher at 26%, and we expect this to reach about 35% in the next 5 years, as democratization of sequencing continues and as research projects increase in size. We have a 4-pronged strategy to strengthen our leadership across these key markets and position ourselves for long-term growth. First, we will deliver breakthrough innovation to remain the NGS platform of choice across research, clinical, and applied applications. We'll deliver end-to-end tests in select areas where we have differentiated skill and scale. And we'll accelerate adoption in existing clinical markets like oncology, reproductive health and genetic disease, And fourth, we'll catalyze new areas like drug discovery, proteomics, and others, seeding future growth. Let's go through each of these. First, we're focused on being the NGS platform of choice across the dozens of research, applied, and clinical markets we serve. To do this, we are relentlessly committed to delivering breakthrough innovations in every aspect of sequencing, from sample to insight, and to continually pushing the envelope to dramatically improve output, accuracy, user experience, price, and sustainability, while also improving turnaround times. Now, these innovations make sequencing more accessible to more labs, allowing us to unlock the elasticity in those markets. Time and again, our new products have helped expand and open up new markets. For example, after we launched the HiSeq X and the $1,000 genome, our customers sequenced more genomes in 1 year than in all previous years combined. Alex Aravinas, our CTO and head of R&D, will talk to you about our exciting new releases and our powerful R&D engine. And Susan Tusi, our Chief Commercial Officer, and Phil Fabo, our CMO, will talk about how these products, including the new NovaSeq X, will unlock the next wave of elasticity in research, clinical, and applied markets. And finally, we have always been committed to our open platform approach, allowing customers and partners to easily and rapidly develop their tests on our platforms. Today, Illumina has by far the broadest ecosystem of third-party applications, including LDT and IVD content. Second, we'll deliver our own end-to-end applications in addition to being the platform of choice across the dozens of markets we serve. We'll pick only a handful of select applications where we look to deliver these end-to-end applications. So how do we decide which markets to target? We're looking for markets that meet certain criteria. First, we're looking for large markets with high growth potential. Second, we're looking for markets where it's possible to deliver a highly differentiated product, for example, through proprietary content. Third, we're looking for markets that leverage Illumina's core competencies. And fourth, we're looking for markets that are strategically important to us. So large growing markets where we can deliver a differentiated product that play to our core competencies and are strategically important. So let me give you some examples. In 2013, we entered the non-invasive prenatal testing market through our acquisition of Veronata. That was the first, and at that time, the largest clinical application of genomics. So big market, potential for growth. Veronata had a very strong IP position, so it was able to establish a proprietary position in that market, which we like. It was strategically important to us, too, because by entering the clinical genomics market, we acquired a lot of capability, regulatory capability, reimbursement capability that we didn't have as an RUO company. And so not only was that important for us to get into NIPT, but it changed us as a company and set us down the path of targeting clinical genomics. And so that's why we did NIPT. A few years later, we entered the cancer therapy selection market through our TSO 500 product. Again, very large market for cancer therapy selection genomic testing, high growth market. We were able to get a proprietary position because at the time, the entire market was focused on small panels, and we went after the 500, the large panel market. And strategically, it was important to us because we knew oncology was going to be the largest market for clinical genomics. And so we wanted to get deeper into the oncology space. Now, in both of those markets, we continued to support partners who built their own tests to compete with our tests. And in fact, we today still make more money from supporting partners, from selling our sequencers to partners that create their own tests than ours. So we've established a model where we can enter these applications and still support partners to compete with our applications. And then we entered the cancer screening market through the acquisition of Grail. Now, Grail also fits those criteria. Cancer screening is the largest genomic application that we're likely to see over the next decade. Grail has a privileged position. It's not only the only player in the multi-cancer early detection space, and likely to be that way for a while, but it has a very proprietary position because of all the IP that it's generated and the patents that are both issued as well as pending. And strategically, it's very important to us because Grail will be servicing millions and then tens of millions of people every year, giving us a front row in terms of being able to innovate for future tests. Now, while I'm on Grail, I'd like to mention a few things. First, we believe Illumina's acquisition of Grail can have life-saving benefits for people who have cancer and don't know about it. We also believe it creates long-term shareholder value for Illumina's shareholders, which is why we did the deal. Second, we believe the Grail acquisition is pro-competitive, and the recent decision from the FTC's chief administrative judge supports this view. Third, as we work through this process, we will always be pragmatic and make the right decision looking forward for our shareholders. And finally, in parallel, as we said, to the appealing the EC decision, we are starting to explore options should we have to divest Grail in the next 9 to 18 months. Turning now to our third strategic area. Our clinical markets are underpenetrated. As a market leader in clinical genomics, we play an important role in driving markets forward, well beyond delivering technology. Now, we do this in a number of ways, partnering closely with our pioneering customers to create successful implementations, from the first population studies in the 100,000 Genome Project with Genomics England to the first national health system rollout of genomic testing and the NHS. We then help share these learnings across our customers through events like last week's IGF. We also help demonstrate the clinical utility of NGS through evidence generation to accelerate reimbursement. We currently have more than 260 studies and research collaborations in the pipeline, including our recent work in Belgium to demonstrate the utility of comprehensive genomic profiling within a national health system. Our market access team is working on increasing reimbursement for NGS globally from 1 billion covered lives globally in 2021 to 2 billion by 2026. In addition to reimbursement, we're focused on establishing pathways for regulatory approvals for genomic technologies. We have reached more than 1,000 IVD EUA product registrations across more than 60 countries. Phil will provide more color in his remarks, and Susan will touch on how our customer relationships support these efforts. Finally, we're working to catalyze new markets, seeding the next big genomics markets in areas such as drug discovery, proteomics, cardiovascular, and neurological disease. In drug discovery, for example, we're working with multiple pharma partners to support greater genomic target usage in drug development, partnering with groups like Nash Biosciences to develop large data sets, and with Deerfield Management to help advance genetic-based drug targets. In proteomics, we're collaborating with multiple players to accelerate the use of proteomics by leveraging NGS as a high-throughput readout platform. This includes our co-development work with SomaLogic on a proteomics assay that's on track to launch in 2024. This strategy has enabled us to deliver strong and profitable growth. For core Illumina, this represents a 14% CAGR for revenue and a 15% CAGR for operating profit in the last 5 years. We've maintained this growth rate even with vast changes in the industry and challenging global macroeconomic dynamics emerging even stronger. The underlying growth for our markets gives us confidence in our long-term trajectory despite current dynamics. We will enable our long-term growth through our innovation flywheel. Now, breakthrough technology innovation is the underpinning of this flywheel. Our innovations have earned us the largest NGS customer base in the world. Now, these deep customer relationships with over 9,100 customers, which Susan will talk about, helps us deeply understand how our customers are using sequencing technology, but more importantly, how they need it to evolve. This closeness to our customers gives us an early look into new applications, and it helps us innovate in ways that anticipate and help us meet future market needs. You can see this with some of the innovations from performance to stability to sustainability that we delivered last week in the NovaSeq X. This innovation flywheel is a powerful competitive advantage, especially when it's coupled with a best-in-class team. On the governance side, we have a world-class board of directors with deep experience, broad and complementary skill sets, and diverse backgrounds. 2 of our independent directors have large public company CEO experience. John Thompson, our independent chair, was recently chairman of Microsoft and previously CEO of Symantec, and Gary Goodhart, CEO of Intuitive Surgical. Francis Arnold, a chemistry Nobel laureate and professor at Caltech, is, as you can imagine, an active chair of our board Science and Technology Committee. We also have deep regulatory experience, including Scott Gottlieb, the former US FDA commissioner, and deep financial experience in regulated markets from board members like Carolyn Dorsa. The board's expertise and strengths across multiple areas of the industry ensure that we have broad perspectives, critical guidance, and strong governance as we develop and implement our growth strategies and advance human health. Finally, Illumina is led by our incredible management team. These leaders represent a wide variety of backgrounds, and each brings both deep technical skill in their area as well as a breadth of experience. I'm going to embarrass a couple of them now, but Alex Zaravanis, for example, has been in the company for over a decade, and we're very fortunate to have somebody leading as our CTO and head of R&D, leading product development, who is both an MD from Stanford as well as a PhD from Stanford in engineering with an expertise in AI. That's a very unique sort of combination to get and is incredibly helpful to us as we plan out future innovations with an eye towards clinical markets. Susan Tucci, our Chief Commercial Officer, another decade-plus veteran at Illumina, is in the position of having built our products before she took over her current role as Chief Commercial Officer. You can imagine there are very few people more passionate about getting our innovations into the hands of as many customers as possible. And we're lucky to have a combination of both veterans at Illumina as well as attract some extraordinary new talent. So Carissa, for example, Rollins joined us from United Healthcare, where she was their CIO and is now our Chief Information Officer. We've also had John Frank join us as our Head of Government Affairs. He joins us from Microsoft, where he was there for over 20 years, including roles like, heading government affairs for them in EMEA and Deputy General Counsel. This strong team, combined with our huge underpenetrated markets, as well as our strategy and innovation flywheel, form the foundation for us meeting our long-term targets. And Joydeep will talk about our long-term targets in his section. So now, please join me in welcoming Alex Zaravinis, who will discuss our recent launches and share more about our innovation roadmap.
Okay. All right. Hello, everyone. Welcome. Just a little bit more about my background. As Francis mentioned, trained as a physician scientist, spent a lot of time at Stanford in academics. Started several companies that got acquired by larger companies. And then I came to my first tenure at Illumina, led a lot of the significant research and development projects there, launched several products. Then spent 5 years as co-founder, leading the development of the Gallery test. And then I've been back at Illumina for several years now. My function, research and development, oversees everything from the very early breakthroughs all the way through development, through product launches, and through on-market support. And it includes all aspects of our technology, so engineering, chemistry, bioinformatics, and software. Really incredible team and really a dream job. Okay. Some key messages that I want to impart to you in this meeting. Look, today we have the leading products. I hope you'll agree after seeing what we presented last week that we're going to have the leading products for a long time. We're also investing in things already that are going to be equally exciting in the years to come. And it's really this product leadership that, as Francis said, has positioned us commercially with such a broad set of customers and the rapid growth. We also want to talk about our R&D leadership, and I'll go more into that. I believe we have the best innovation engine in the world, both in the people, our culture, our processes, and products like the NovaSeq X really speak to that kind of success. I also want to talk about, I'm going to go into more depth about how we think about developing products to create long-term differentiation. And it's this type of differentiation and very hard to replicate technology and products that have given us these leading products that customers prefer and that will prefer long-term. The last point I want to also touch upon is how we think about getting the most out of our R&D dollars. And we spend a significant amount of shareholder investments on the innovations, and we think deeply about how to make big innovations that we can leverage across multiple products and kind of sell multiple times. So I want to share that thinking with you. Okay, you're going to hear these numbers several times about the unique position we are in with so many customers, so many installed systems. From my point of view, this gives us the opportunity to really see what's going on in a way that perhaps no other company can, right? So I and my team, we regularly meet with our customers. We understand the challenges they're having at times, where they want to go, what are the new applications, what are the new features. And then we feed all that in a very customer and market-backed way into how we think about the innovations that will be exciting products that our customers will want to buy, that they'll want to adopt in greater numbers. These key stats around R&D are really important. So have over 2,200 people in our core R&D team. And again, that's kind of every major discipline of science, engineering, and software. We produce about 400 invention disclosures a year. And that's a testament just to the amount of, the incredible amount of innovation that we're constantly doing. You know about the hundreds of thousands of patents, right? And again, that's also a demonstration of the wide use of our technology. We have over 1,000 issued patents and then many, many more pending applications. And so we work hard to protect all of those innovations that we're constantly producing around our differentiated products. A few more things. One is we think a lot about the culture of the R&D team, the composition of it. Very proud of the diversity, which has gotten significantly better in recent years. This has allowed us to draw on a deeper talent base and really an advantage in how we do R&D. We have very low turnover relative to our industry. I think that speaks to how exciting a place Illumina is to be when you're working on new technology and developing the new science. We have about a 90% acceptance rate in people coming to Illumina when we give them an offer, right? And when I ask them about why they come, and this is very conscious in how we think about it, if they want to work on the products that are going to change the industry, that are used by the majority of people, they want to have access to the resources to make those big game-changing investments. And that's what you get at Illumina. And that's why I think we get the best talent in the industry. We retain it. We also have a very senior team. I have about 20 vice presidents and distinguished scientists and engineers that help me lead the function. The average tenure of that group is 10 years at Illumina, and then many of them even have broader experience in the industry. And so the benefit of that is these are people who literally built everything, both at Illumina and the largest parts of the genomics industry. So that is a unique asset in how we think about where we're going to go that I think is unparalleled. Okay. I want to give you some insight into a few different ways that we think about how to innovate to create this really enduring differentiated value. The first is what we'll call enhancing value beyond just cost per sequencing or dollar per gigabase. Now, I don't want you to leave for a second thinking that we're not committed to that. As you just saw, we did a huge price reduction in the cost of sequencing, and that is through massive innovation, right? Over time, we are exceptional about learning how to scale technologies and make innovations that allow us to produce the technology at ever-decreasing cost, and then we can pass that on to our customers, and then we enjoy the elasticity of that. But we think much broader about that. And I'm going to use NovaSeq X as an example to talk about this. So again, I believe we have the underlying technology and infrastructure to produce sequencing technology at lower cost than anyone else. That's a huge advantage. But beyond that, we also scale, right? So that allows people to do much bigger projects, much bigger cohorts, right? And you see that in NovaSeq X with a 3x increase in output and twice the speed and the cost reductions. But because of our experience, our deep experience working with customers, we understand what can slow them down, what can limit adoption, what could lead to inefficiencies or waste in their workflows, and we address that very explicitly. And NovaSeq has a huge amount of technology packed in to address that. As an example, we do clustering on board, right? One of the few instrument providers that does that. We also have addressable lanes. It makes the fluidics more complex, but it gives tremendous workflow flexibility that customers value. It used to be that you would have to manually load individual sequencing lanes, and if you wanted to do large numbers of samples, there was a lot of complexity up front of the sequencer. We've automated that, and now it's eliminated from the customer's hands, and they love that. We've also made the instrument incredibly easy to use, and after the session, we're going to spend some time for those who want to get up and close with the instrument, and you'll see how beautiful and elegant it is. And what all this means for our customers is that they can do more sequencing, it's easier to use, and that's great for the discoveries, that's great for their patients, and great for our business. I also want to talk about us as a trusted partner to customers, right? So we have 24/7 support through Susan's team. It's worldwide, and we support everything, right? So it's not just the instrument, but if you have a problem upstream, you have to call some other company, or you have a problem with the software, you have to call yet another company, figure out how to put all that together. No, with Illumina, when you're part of our ecosystem, you get supported across the board, and we do it quickly so that you can get back to sequencing. And again, I think having the full portfolio of products and having that kind of service is something that our customers value. And we're committed to that, even in the research and development group by supporting that. We also have a huge menu of applications. And so whether or not you're doing tumor sequencing or you're doing germline sequencing or single cell or even spatial, we support that, particularly on the software side. And so that means that a customer who wants to get into these new areas, they get that menu day one, right? And they get it from us, and they don't have to pay extra for it. So this, again, allows them to get into new applications very quickly. And then the last thing I want to touch upon is the sustainability and how important that is to customers. We made, because of our innovations and the uniqueness of XLEAP chemistry and our packaging, we can now ship ambient. Huge value to customers there, not just in achieving their ESG goals, but dramatic reduction in the complexity of how they can use sequencer and cold chain logistics. I'm going to use NovaSeq X to also talk about very specific technology innovations and then how those translated into big product benefits. The first is the ultra high density flow cells. So the new flow cells for NovaSeq X are over 300% denser than NovaSeq 6000. And this is why you now get effectively 3 NovaSeq 6000s in a single instrument because of these incredibly dense flow cells. Now, to make flow cells this dense, the technology and manufacturing processes did not previously exist. So it was a very long-term research, ultimately development, and then scaling operation to do this for the product and to meet the demand. So again, an example of something didn't exist, very challenging to do. We made huge investments, had hundreds of people work on it. We not only made the breakthroughs in the basic science and materials about how to do that, we then figured out how to scale it and how to automate it. And now with those increased densities and the reduced costs, we can now bring that to customers and pass it on to them. Similarly with XLEAP SBS, right? The previous generation of SBS has been amazing, and it built our industry. And improving on it was no small feat, right? And maybe a testament to that is all the participants, new participants in the market that have had to use our original version of XPS. But our team, they went back, and this is the team that invented SPS and developed thousands of new chemical structures and discovered an SPS that's even better, more stable, and that is what allowed us then to use it on the high-density flow cells to achieve ultimately longer read lengths, to achieve higher quality and ambient chemistry. So we're very proud of that innovation, and customers are extremely excited about it. There's a whole new set of compute hardware and algorithms in NovaSeq X. So we have large teams that we've been developing this for years also. It's hard to overstate how important this is to customers. Number one is with all this additional data, you would have a big problem processing the data, which would make the run lengths long, delaying when customers could get their outputs. But we've solved all of that through those innovations. So by having accelerated hardware, better algorithms, now you're actually getting your answers faster, even though the system is producing more data. We also included Dragon on board, and so now all the secondary analysis, for the first time ever on a high-throughput sequencer, is included as part of the cost, and with a huge benefit in speed. A few other things, and again, when we innovate, for example, in a high-throughput sequencer, and you make a better chemistry to fully take advantage of that new engine, you then have to invent and innovate across the board, and that's how we think about it. So when we did these innovations, we had to do them all so that we could have a product that could fully take advantage of any individual one of them. Last one I'll mention is the optics. And again, no cameras and no objectives and materials existed that could scan at these speeds to create the 300% increase in output. So multiple inventions, multiple years working with leading partners on the materials, the optics, to create those. And again, proud to bring those to the market but before anyone else so that our customers can benefit from that. So again, I hope you're appreciating here that this is not easy to do. It takes a company like Illumina, which is unique in terms of the scale, the number of people, our ability to make these investments over years and years, make those breakthroughs, protect them with IP, and then also figure out how to commercialize them in a robust way. I want to take a little bit of time to talk more about the benefits of the ambient ship, the environmental benefits, and then also the customer logistics. So after Francis and I announced this product at IGF, I had several customers come up to me, some of them in tears, about how excited they are about this, that they thought this would never come. They thought it was, a scientific impossibility. And what it means that now, instead of filling dumpsters with, cardboard and waste and having big teams that have to manage all of this, a single person can pick up a 10-pound box from UPS. They can load the world's most powerful sequencer. They can process, over 100 genomes, and they don't have to fill up a landfill, and they don't have to manage complex cold chains. The other thing I want to point out is that this is impossible to do with the original version of SPS. The chemistry is just incompatible with ambient ship. So without XLEAP SPS, its new bonds, and many other new inventions, you can't achieve this. So again, long-term dream of ours to do this, and we couldn't be more thrilled about finally getting this into customers' hands, and the initial response is just over the moon. Now, these innovations, as I mentioned, we leverage across multiple platforms, right? And by making far-sighted investments, very big, not incremental, very big leaps in technology, and then doing it in a way where we can use it in multiple platforms over time, that gives us a big return on our investment dollar. It also it's one of the reasons we can do more products faster than anyone else in the industry because of this concept of leverage. And we think about that day one in research, right? We don't want to make something that will have to be done in an artisanal way. We want to make something that we can scale through semiconductor manufacturing, right? We work with vendors very early on, for example, with our operations group, around thinking through all of that so that when we get to the point we want to scale it, we can do it rapidly, we can do it at low cost, and it has the flexibility to use for years and years across multiple product lines. A great example of this is bringing the new XLEAP chemistry to the NextSeq 2000. So this wasn't an accident. This was around, as we were launching NextSeq 2000 several years ago, thinking about, knowing about what we had in development, even though it wasn't perfected yet, and saying, look, there's going to be an opportunity for customers that'll be really exciting, where when we have a chemistry and a flow cell technology that can go even denser, we could offer it to our NextSeq 2000 customers. And this is when you're thinking over 5-10 years, and you're working on all the segments thinking very deeply about this. So NextSeq 2000 has optics and it has engineering that allows it to go at launch to a density that didn't even exist in terms of the chemistry that was needed. And so now, with the investments in perfection of XLEAP SPS and NovaSeq X, we can now bring that to the NextSeq 2000. And this is super exciting for our customers, that they're going to get an even deeper roadmap through their existing investment in this exceptional mid-throughput platform. I want to remind you also that we've gone to longer read lengths with SBS, both with the original SBS, for example, our recent announcement of the 600 cycle kit for NextSeq 2000, and that's 2x300. For those customers who wanted to do much deeper sequencing of TCRs and BCRs, of T-cell receptors, B-cell receptors, this is a great product for them. To my knowledge, this is very difficult to do on any other type of SBS, which is why you'll see them tend to be limited to shorter read lengths, but a unique capability. XLEAP SBS has even more of this potential to go to longer reads, and we're excited about the roadmap there around that. I want to highlight another benefit for customers when they buy one of their systems, which is they've come to appreciate that they're getting a great product at launch. Then they're getting extensions, which give increased output over time, reduced cost over time, and now we're even showing that they even get things that were beyond the roadmap that they expected, which is longer read lengths or even access to a next generation of chemistry. We also had another important announcement at IGF was the launch of our NovaSeq 6000 DX. This is the world's first FDA-cleared high-throughput sequencer that's IVD compliant. A great product for those customers that need or prefer a cleared instrument, either in their geography or their type of lab environment. Very good initial response to this. It's also a product that has improved features for these types of lab environments. It has streamlined workflows, it has additional automation, and has the ability to switch between RUO and DX. So a lot of thought here about how to make about making this product exciting for customers that want the DX version. One other important point is that when we develop our instrument platforms, we develop them in a way that we can make anything DX. Okay, so it's additional cost and expense to do that, but you have to do a lot of upfront work in how you're developing the system, how you're documenting it, how you're qualifying it, just a different level of quality. And so we forward think about that so that for those segments where it makes sense, or for particular platforms or at the right time, we can bring a DX to market. That required a huge investment in infrastructure to be able to do that. Your average RUO instrument cannot be turned into a DX instrument. Want to point out that now with the NovaSeq 6000 DX, we now have the only portfolio of a DX instrument in every single segment. So the lower throughput desktop, the higher throughput desktop, and now the high throughput. For customers who are using the DX platforms, this is a great opportunity for them because it allows them to scale DX workflows, and stay on a cleared instrument the whole way. Okay. A couple other areas that are very important when we think about innovation, creating more value for customers. We call this delivering the kind of the best and most complete view of the genome. So if you take a step back and you think about the people who use our machines, whether or not it's for scientific breakthroughs or clinical applications, in the end of the day to them, it's about getting the insights. They learn something new about science, and that gives them the opportunity to publish and share it with the world, or they found something they can use to help a patient, get them on a therapy, give a parent an explanation for a developmental disorder. And so the easier that is to do, the faster it's to do, and the more of those insights, well, that makes the technology more valuable. And then over time, it allows them, our customers, to justify making even bigger investments in our products and our technology. So we've been working for years to automate this and add more and more value, to cover more of the genome, to cover more variant types more accurately, excuse me, and to add more medically relevant ones. I also want to touch on the amount of data coming off our system. So in 2021, it was 280 petabytes. To put that in context, that's about 100x the complete Netflix library of content. This is every year, and in fact, it's almost doubling every year. So it's a lot of data. And again, when we think about our customers, this is a huge call to action, right? Which is they want innovation here. And they value innovation, and it affects not only what they can do with our sequencers, but it affects how they think about which sequencers they want to buy. Right? So they want this to not only stay the same, they actually want it to get faster, even though there's more data coming out. They want it to be more accurate. They want to be able to do it with fewer experts, and they want to get to the insights quickly, not the intermediate complex data processes. So we now have a complete technology stack to do that all for them. And in the case of the NovaSeq X, there really is a supercomputer on board now. That's never been the case in the past with Dragon 4.0. With this informatics, we've now increased the accuracy of the genome to 99.83%, and that's in the FDA Truth Challenge Reasons. And this is independently, tested in those FDA regions in their competitions. So this makes it overall the most accurate genome, right? And that's short read, long read, the most accurate overall. And then the graphs on the right are to highlight the massive improvements we've made to speed and cost reduction. And this is through accelerated hardware that's much more efficient at processing this type of data. So it requires a lot of engineering to do it, but once we do it, then if you're using Dragon in our unique systems, then you can get these answers much faster, and you can get them at much lower cost than anywhere else. And that goes straight to the benefit. Those benefits go straight to our customers. Also want to highlight Illumina Complete Long Reads. So at IGF, we talked about the fact that there is now a platform where you can get both short and long read information, and that really is true. So Illumina Complete Long Reads is not a synthetic long read technology. It's a way of preparing the DNA, then going through one of our sequencers like a NovaSeq 6000 or a NovaSeq X and getting true long read information. These are complete contiguous long reads. It's the original single molecule represented. There's no strobing, there's no dropout. And for key hard regions of the genome that have historically been hard, which is about 5%, you get the information you need or we're looking for to make a complex rare disease diagnosis or new science that you're interested in. Now, not only is it incredible that you can do this on the existing platform you own, or if you're buying a NovaSeq X, the workflow is actually also incredibly easy. It's a single-day workflow, very straightforward, doesn't require any specialized equipment. So we worked a lot to make it really easy to do. It also doesn't use a lot of DNA. Most long-read workflows, legacy ones, require a lot of DNA, very limiting. We heard that from customers. We wanted to make sure that this was a low-input workflow. And importantly, the accuracy is even better when you combine this with, let's call it the standard, whole genome. And so now we're at 99.87%. So now, hands down, the most accurate human genome in the world, and one you can do at reasonable cost, and one you can do at scale. Now that 0.87, that 0.83 to 0.87, that may not sound a lot, that additional 0.04, but what that represents is, in some cases, a very particular gene that could be relevant to a pharmacogenomics study. It could be a child with a developmental disorder, and this gives you insight into that, say, repetitive region. So not a lot in totality of the genome, but tens of additionally important genes for certain customers and certain applications. I want to talk about another aspect of innovation, and that is we think a lot about not only making our existing and still early applications successful, like tumor sequencing, like whole genome sequencing for rare disease, but all the new applications that people are developing, but that use the sequencer kind of as the center of the genomics universe, right? And we've come to appreciate that our sequencers, as we increase throughput, as we reduce cost, they're really a universal biology information machine, because they allow you to count billions of molecules very accurately. And by attaching those DNA molecules to other things, you can do pretty much anything, right? So you can do proteomics, you can do spatial sequencing. There's really no limit to it. You can see the applications here in our ecosystem. So all the ones that, again, we're working hard to continue to grow with customers like Somatic and Germline. We've done a lot in transcriptome, but still a lot of new exciting stuff there. Single cell spatial proteomics, epigenetics, gene editing. So this whole ecosystem of new applications. And each one of these, we're very close to partners and customers who are doing these. We have, in some cases, joint commercial arrangements to accelerate these, and make it easy for customers to adopt these and scale them. And then in some cases, for example, proteomics, we appreciated we could do something unique by making a full integrated workflow ourselves. And so that's the partnership with SomaLogic. Now, we're very excited, and you can imagine running that new workflow on the NovaSeq X, for example, where you could run many hundreds of samples on a single run to do proteomics. You could do 10,000 targets with 10 logs of dynamic range, and you can do it at the lowest price point, right? So we think that will really help take proteomics to where we've brought genomics. The last is end-to-end workflows. That's another place where we create a lot of value for customers. And so through our innovations and investments around the sequencer, we've created end-to-end workflows for some major applications, right? So today we have automation software. We have laboratory and information management software, which customers need if they're doing large sample volumes, particularly in clinical environments. We have the world's leading secondary analysis, right, through Dragon, which has won all the FDA competitions, not just in germline, but now in somatic analysis also. And we have an ever-growing portfolio of tertiary analysis, which helps customers with that last mile of going from variant calls all the way to reports and all the way to interpretation. And then you can see these horizontal here where we take advantage of our investments, right? So whether or not it's rare disease or oncology, infectious disease, we use these components and then we make complete workflows. Over 2,600 customers have adopted one or more of these complete workflows. And so the feedback from them is they want more of this. They want more of, they can buy a single solution and they can get into one of these applications very quickly. They, they really, want to avoid scenarios where they have to get these pieces from different vendors, kludge them together, then make them work, have a much larger staff to do all this kind of construction and piecing these things together versus applying their R&D dollars or their other dollars to running samples and doing what is perhaps their core business. So again, we think very carefully. It's a large investment when you do one of these about where it's big enough, where we can do something unique enough to create a lot of value. But when we do, it's tremendously appreciated. Also point out here, it's not only a way to kind of drive more business to our sequencers. It's also a way to create more value, right? These other components are things that customers will also invest in. Okay. All right. Well, I'm going to end there. Hope I was successful in helping you appreciate that innovation is core to our success, right? Because innovation gives us enduring product leadership, which we've had historically. Which I think with products like NovaSeq X, the NextSeq 6000 DX, bringing XLEAP to the NextSeq 2000, Illumina connected complete long reads, our commitment to again, product leadership for years and years to come. But I'll give you some insight into how we do that by having the world's largest and I think most successful culture of innovation in the space, how we think about making investments that can be leveraged over many years, unique technology components that are protected by significant IP, how challenging these things are to do, and just our repeated success in making these breakthroughs and small miracles happen, and then bringing them to customer hands. And then last, that appreciate all of your support and the investor dollars that make this possible, and that we try to be very good stewards of how to use this money to do these investments, not only in a way where we make breakthroughs and great products, but we do it in a way where we're very efficient about our use of this capital. All right, I'll stop there. And I'd like to introduce Phil Febbo, our Chief Medical Officer. Thanks, Phil.
Well, thank you, Alex, and it's great to be here with all of you and have the opportunity to talk to some of you. Some of you I've worked with almost a decade in my two roles in industry, and you know me quite well. But for those of you who don't, I'm Phil Febbo. I'm the Chief Medical Officer. I'm a medical oncologist and physician scientist who spent the first 20 years of my career as a physician in academia, first in Boston at the Dana-Farber, where I did my training and was on faculty as a physician scientist, bringing genomic insights into understanding the biology and clinical behavior of prostate cancer. I moved to Duke University for 6 years and then to UCSF, where I finished my academic time as professor of medicine and urology, and transitioned to industry because I really, I've been my career is really focused on bringing insights from a cancer's biology and genetics to care. And whereas in academia, the initial insights and translation is fabulous, it's really moving to industry. I felt like I could have the impact and really make sure that these insights translate into changing care and improved care. So I spent first 5 years of my academic career at Genomic Health as Chief Medical Officer, helping build the evidence and reimbursement for proprietary expression-based tests with their Oncotype DX test, and transitioned to Illumina because at the end of the day, I am strongly biased, and you could argue I'm biased because I'm at Illumina, but I'm actually at Illumina because I am biased that individual's genome and genomic insights should be foundational to health and healthcare. And that's what drew me to Illumina. That what makes me so excited to be their chief medical officer. I also serve on the foundation boards for the FDA and the American College of Medical Genetics and Genomics. So let's get into it, because at Illumina, we've built a clinical playbook. Alex has spoken about our incredible technologies, and increasingly, those technologies are being brought into the clinic. And what I'd like you to take away from today, as far as, my key messages are talk about that clinical playbook and walk you through the incredible team and infrastructure we've built to deliver to clinical customers, and demonstrate that investment is a good use of investment dollars to drive our business, and also talk you through where we see our key clinical segments driving the business. Francis already talked about some of the market opportunity, and Joydeep will follow up on that, but I'm going to walk you through the key clinical segments and where we see real opportunity to drive an improvement in health and healthcare. That's my role as Chief Medical Officer, but it's also a big driver for our business. So let's talk about the clinical playbook that we've had built and that we've really accelerated over the past 5 years since I've been at Illumina. You've heard about our products, and our products are foundational to our playbook, and I'll walk through why that is. Not only our sequencers, but the fact that we're embedded in all the clinical leaders that are bringing genomics to healthcare, and that gives us insight that we need to have both sequencers and menu on those sequencers. But then again, you also have to have the right to operate. So we'll talk about moving to regulatory approval and the infrastructure we've built to have that right to operate globally. And then you move to the why. So, of course, it's one thing to have a sequencer and be able to do testing, but what's the utility? What is the benefit to the patient? What is the benefit to the healthcare system? And once you demonstrate that benefit, you drive to reimbursement so that our customers move genomics from a cost center to a revenue-generating activity, and that really further drives our business. And we then partner with them broadly to ensure that every patient has access and that these tests are adopted. This evolution takes time. It takes time in each segment with each type of testing. It's furthest along in oncology with therapy decision, comprehensive genomic profiling, now the standard of care in the United States and Germany and many countries for patients with non-small cell lung cancer. That's because the providers are there, the clinical tests are there, they're available, they're reimbursed, and now they're being progressively adopted. We've seen that same evolution in NIPT, and we'll touch upon that. Incredible success, the fastest adopted and penetrated diagnostic test ever. But we're earlier in genetic disease where we have evidence, but we've just recently performed a study with Optum showing that whereas there's evidence and guidelines, only 5% of newborns suspected of genetic disease actually get genetic disease testing. So we have more work to do there. And with the pandemic, infectious disease is moving out of the research into surveillance and indeed into clinical testing. So I'll walk through some of those But let's start with the playbook. And of course, we're very proud of the sequencers that we have across the markets. And, just like research, in clinical labs, one size does not fit all. And so it is incredibly important as a clinical lab begins as an entity and then scales as an entity, as Alex said, that they have a company that can grow with them, that can support that scale, that has products across. And of course, we've built our products focused on research use only, and the vast majority of our clinical business right now is supported by our customers taking research use only products and making their own tests. But the evolution in healthcare is moving towards a more regulated and an environment where you have to move on to cleared or approved products. And that's why we're really proud to have built the MySeq DX initially, the NextSeq DX, and indeed the announcements of the NovaSeq on Thursday that Francis and Alex made is an incredible event because it completes our low, mid, and high throughput portfolio and to both serve RUO and diagnostic machines. But what are companies doing on these machines? And I think starting with on the right side of this test, the laboratory developed tests have been a model for innovators, and we are the platform of choice across all the clinical innovators. We are embedded, they're using our sequencers, and we will continue to be the platform of choice. And I think the announcement of the NovaSeq X takes it to the next step, offers them the next level of scalability and for their business. The other element is that as we've become more global and as genomic testing has moved outside of early adopting countries and laboratories, it has to move to more content and in vitro device content. And given the diversity of the world and the different evolutions of healthcare systems across the world, one company cannot produce all that menu, all that clinical menu. So we've taken an open platform approach and worked with innovators to create IVD content on our platform and provided the support so that they could do that. Of course, in areas where we see our internal team and our knowledge of sequencing can further accelerate and accelerate a field, and where we can participate more in the value chain, we do develop our own content. So we've done that in oncology with our very first cutting of our teeth with the Praxis test, working with Amgen for RAS testing. And then, we've recently released the TSO Comprehensive in Europe under a CVIDV mark. We've released content in genetic disease, reproductive health, and infectious disease during COVID with the COVID-seq, which has been a backbone of genomic surveillance across the globe and accounts for between 40 and 50% of submitted sequences to GISAID. And the importance there is once with our established install base and with our growing established install base in DX machines, it's very clear to pharma partners that if they want to identify the most number of patients with a variant that can benefit from their drug, Illumina becomes a partner of choice. We haven't been that traditionally, but over the past 5 years, we really built the teams and the infrastructure to support that, and we have the products that are foundational upon which the analytic products upon which CDX claims can be placed. So we have the product as far as the sequencers. We're growing the content through partnerships and directly, but you have to have the right to operate. And over the past 10 years, Illumina has worked to make that transition from a strictly research-serving company to a research and clinical serving company. And it's really been working with leaders of product development, Susan before Alex and now Alex, as well as our leadership in our operations, Kevin Pagels, our Chief Operations Officer, together with medical, to have a culture of quality. And everyone comes to work at Illumina dedicated to quality. But when you're moving into the clinical markets, you have to document that commitment to quality through processes, documentations, and certifications. And so across the globe, we have ISO certifications, MDSAP, IVDR in 3 labs in the United States, in the UK, Netherlands, and Singapore, so that we have access to global markets through those certifications and that commitment to quality. And that becomes incredibly important. And it's resulted in some of the numbers that Francis talked about with 72 approvals from regulatory authorities, over 1,000 products that are registered across the globe, giving us that right to operate. So that's the how. What about the why? Why should these tests be ordered? And that's the clinical utility. And as Chief Medical Officer, that's where I focus. Like, how do we demonstrate that these tests improve patient outcomes? And in today's super strained health systems across the globe, you also have to say, not only they help the individuals, but they help find healthcare efficiencies. And so you do that through evidence. The coin of the realm is, what is the evidence for these peer-reviewed journals that there's clinical utility, that patients benefit. So in our key segments, we have hundreds of papers, over 800 in reproductive health, oncology, genetic disease testing, and infectious disease, focused on our products and our technology improving outcomes. This is across the globe. We're not just focused in the United States, obviously. We look because in each environment, that utility equation is not the same. And payers in specific healthcare systems want to see evidence within their systems for the populations they cover. And we've done that in spades. We're highly efficient with this. We're very careful with the dollars that we spend here. And we collaborate with the best medical societies, the best academic centers. There's incredible interest in using our technology to understand how to improve outcomes. And we can partner very efficiently to generate studies that are incredibly powerful. And I want to talk through just a few of those. So for comprehensive genomic profiling, we've worked with Amy Compton-Phillips in her role at Providence, formerly known as Providence St. Joe, and worked with her to facilitate their commitment to internalize CGP and then offer it to every patient with advanced cancer in their system. This is an integrated healthcare system. They're in charge of that patient, of all the costs associated with that patient. That interaction, that activity, over 10,000 patients have received that test. We have 8 abstracts we've worked together. I was just at ESMO where we had the opportunity to present a few more. 3 manuscripts in development that'll drive the evidence showing how it's good for patients, because you identify, for example, every patient with an NTRK fusion where the benefit of getting larotrectinib or entrectinib is incredible. as well as for the healthcare system efficiency to internalize that and have that so they're not doing test send out. Another study is the Ballet study, and Francis mentioned this. This is our interaction in Belgium. And what's incredible is that Belgium is a single-payer system, and we've worked with 12 hospitals and 9 Belgian NGS labs to provide comprehensive genomic profiling in collaboration to 960 patients with solid tumors. I met with Birgit Meis, the PI of the program, in her small center outside of Brussels just about 6 months ago. And it's incredible. She has a real-time dashboard leveraging all the informatics that Alex talked about. So in real time, she can tell me how many patients have specific variants, NTRK, RAS mutations. That's incredibly powerful because not only does it make sure everyone gets standard of care, but now they're a great site for additional clinical trials where pharma companies increasingly want to know that these centers already know what variants their patients have to facilitate accrual. In the genetic disease, we saw a big opportunity to move from single center evidence where it was becoming more and more clear that children suspected of genetic disease who were in the NICU and sick benefited from whole genome sequencing rapidly, work from Stephen Kingsmore, Mercy Children's in Kansas, and others. But there was no level 1 randomized controlled study showing that benefit. And so we took on that with key leaders like CHOP, Children's in Boston, and we performed a randomized controlled study, the first one for NICU patients, demonstrating that whole genome sequencing doubles the diagnosis when provided immediately and doubles the opportunity to change management and doubles the change in management when provided earlier. Highest level of evidence to continue to drive towards coverage. So those are some examples. We also extended from our Baby Bear activities in California that opened up WGS for infants in California. We're doing that in Germany with the Baby Lion. We're doing that in Israel with Baby Bambi. And we also have a growing team to collaborate within China and perform collaborations, bringing whole genome sequencing and insights around that to that country. So they're just a few select. We have hundreds of these collaborations that are very efficient and performed through our teams. And why do we do that? Well, at Illumina, our business grows together with our clinical customers. So while we drive our business through driving the success of our customers, and that evidence drives towards reimbursement. We're very proud that we've played a significant and strategic role in getting to the 1 billion lives covered. We're committed to getting to 2 billion lives covered to continue to help our customers. And we do that through meaningful collaborations with key payers and evidence bodies across the globe. And we're just listing some of them here. That's because, as Sean Tunis says, when you have coverage from one payer, you have coverage from one payer. Achieving reimbursement is not a passive, it's an active game, and you have to engage, you have to provide the dossiers in the language that they understand to move it forward. And we rise all boats. Certainly where we have tests that are directly affected, we benefit disproportionately, but because our sequences are in all clinical leaders, by raising reimbursement, we benefit from that. And when there are particular challenges, we'll engage in risk-sharing projects like we did with Harvard Pilgrim, now known as Point 32 Health, where we said, if you opened up NIPT to all comers, and we'll measure the health utilization during the year when you open it up, not just to high-risk pregnancies, to all pregnancies, and we'll look at the year before where it was just used in high risk. We'll backstop any increased financial burden in that utilization. What happened is there was a slight incremental cost that we covered, but when they folded into their lives covered, it was only 2 cents per member per month, which is one of the measures they used. And what their decision was to continue that coverage because they felt the quality and the utilization supported its utilization. And that's where we dig in. We have creative programs like that across. And who's doing this work? And whereas Alex talks about the 10 years because Illumina has really built the sequencing world, so we have people who've built this, Illumina has entered the clinical world because of the transition of our tests to the clinical utilization. And we've drawn talent from clinical leaders across the industry to build a medical team that is now 1,000 strong across the globe and driving the incredible work that we've talked about as far as engaging with collaborations, driving evidence, bringing dossiers to payers, bringing applications for registrations in regulated areas, and also partnering with Susan's team that you'll hear about in commercial, Catherine Reeves' team, our chief marketing officer, to work as a team with our clinical customers. It's taken a commitment on Francis's and all of our part to build that investment. Because each time we're choosing to build someone on the medical team, be it a medical affairs person engaging with collaborations or peer-to-peer education, or regulatory person in a region working on those applications, you're choosing between boots on the ground selling and this support. And that's why we work so closely to make that decision and have built over time. It doesn't happen quickly. Money doesn't solve it because they have to come in and be integrated. And it's really a pleasure to lead this incredible team as we drive our clinical business. But as we transition to segments, you'll have to just recognize, as a medical oncologist, I think about the bigger why. And AJ is an incredible person, and it's someone I've gotten a chance to get to know, and we featured him last week at our IGF, and I've had several opportunities to talk with him and interview him and meet him. He's a 40-year-old gentleman. He's now close to 50, but non-smoker, started having some throat clearing, cough, went to doctors when there was a little blood in his mucus, and that really scared him. Subsequently went through diagnosis, chest x-ray first, suspicious for mass. CT had metastatic non-small cell lung cancer. He was shocked. He's not a smoker, takes great care of himself. How could that happen? Of course, 10% of lung cancers are in non-smokers, but he didn't know that. And he did not have enough tissue for comprehensive genomic profiling at diagnosis, but he did have cranial mets, and they were limited and resectable. And together with his doctors made the hard decision to get a craniotomy and those mets resected, because that's one path of minimizing the impact of those, but also because more tissue is needed. So the tissue was used for comprehensive genomic profiling, and he was still recovering from the craniotomy when the results came back. And it's really interesting. His doctor gave him a rather robust hug, which was a little uncomfortable, but they had found a ROS fusion, which can be treated with crizotinib, and that was 12 years ago. For those of you who know, the average life expectancy for someone traditionally, when I trained, was with non-small cell lung cancer, cranial mets, 9 months. He's alive 12 years later because of matching his disease. It was really interesting when I send a note to him saying, look, I'm going to be talking to our investors. I'd like to share your story. Is that okay? He said, and I quote, "You and Illumina are absolutely welcome to use any aspect of my story. Illumina is why I am here today." So that's the why. And that's the opportunity we have to impact lives. And we've chosen segments that we see the biggest impact to patients and the biggest markets: oncology, reproductive health, genetic disease, infectious disease. And we're going to walk through those. Francis has already touched upon it. Joydeep will go into it. I am Chief Medical Officer. I focus on patients, but I am an officer and have to be a good steward of our company. And these are incredible markets, so that by driving improvement in care, we're driving company growth. Oncology is the biggest opportunity, and a lot of what I've talked about so far with AJ and others is using genomics to inform treatment decision. And we've seen the movement, and I'll talk a lot about this at my last slide, a movement from single genes to panels to comprehensive genomic profiling, and that's where we are for standard of care in the United States, Germany, Netherlands, Belgium, and a growing number of countries. We've had incredible early innovators build that market using RUO, and now it's maturing so that increasingly they're looking for IVD products to decentralize and get into regions across the globe that aren't well served by LDT. Now, while that's the most mature market, it is not the biggest market. The bigger markets are in the screening, and Francis spoke about our enthusiasm for screening. If you were at IGF last week, that is going to be using sequencing for early cancer detection through multi-cancer early detection tests will be one of the ways we achieve the moonshot's goal of having mortality in the next 25 years. Earlier diagnosis will save lives, and it's incredible that we can now pick up on the cell-free DNA signal in circulating in blood to detect cancers earlier, move away from just screening for 5 cancers to as many as 50 cancers. That is an incredible market, and if you look at the basic science, it's very clear sequencing is front and center. It's going to be the lion's share of every product. It's certainly the foundational to Grail's product gallery. And for those that are moving into single-cell screening, like Freenome for colon cancer, moving to blood-based testing, it's all about sequencing. Monitoring is another very exciting area that we'll see grow over time. Natera's been out there doing whole exome on the tumor and then doing a specific PCR-based assay. But as we have breakthroughs like the NovaSeq X, as the cost of sequencing comes down, it's just math and statistics that comprehensive sequencing of the tumor and the cell-free DNA will provide an operationally efficient path to diagnostics that are driving monitoring after definitive local therapy and recurrence. And those are going to be major impact on the care of oncology patients. And IPT is another more mature market. And Francis has walked through how we participated by buying Veronata, by performing a service, using that service to build a product like VeriSeq and IPT, and using data that we've helped through collaborations to build evidence and then driving reimbursement. So if you look at reimbursement, very positive in AMR, EMEA, and China. APJ is a little behind. There's a lot of out-of-pocket utilization and payment, but we're seeing really good activity in Japan, South Korea, where there are already mechanisms where there is some support, but it's not nationalized reimbursement, so we don't count it. We expect to move in that direction over the next few years with our team. And Germany is a great example because we worked very hard with the authorities in Germany to get to both reimbursement and, super important, drive to a reimbursement price that could help our customers in region have a meaningful business. And we achieved both. We achieved the reimbursement decision last year, and this year we had the price set that was very favorable. Importantly, also in the tender, by being engaged, we made sure that the requirements for reimbursed tests met expectations for patients, but they were also perfectly aligned with our product, VeriSeq version 2. Genetic disease testing is a really exciting place where this is the opportunity, and with the NovaSeq X, we will see the genomes becoming foundational to human health. As we stated, it started with children suspected of genetic disease and demonstrating whole genome as the best way to identify the variants causing their disease. We have health economic studies showing that a whole genome at time of birth is not only best for the children, as I talked about NICU-seq, it's also the most economic. It avoids a series of testing and delays in management that puts a big burden on healthcare systems. We will see a transition from people suspected of having genetic disease, both NICU and pediatric population, transition to what are the genetic drivers of common disease. Cardiovascular is first up. We know that there are indeed Mendelian genes that drive risk of cardiovascular health, be it cardiovascular disease, atherosclerosis, cardiomyopathies, arrhythmias, and increasingly people are incorporating that into assessment of cardiac patients. Polygenic risk scores are becoming incredibly exciting because you can identify 5 to 10% of patients with no other history of cardiovascular disease who have as high a risk as an individual who inherits a mutation in familial hypercholesterolemia genes. That's just the start, because when you look at where the pharma industry is going, if you ask the question, how many drugs in development have a variant in the germline that are needed in order for that drug to be beneficial? We looked at this deeply. There are 38 trials in cardiovascular disease, 66 in neurology, 62 in metabolic, and 38 in auto. That means that just like oncology, where therapy has moved from all comers to identifying groups where therapy can be very beneficial based on identifying a variant, that's moving into genetic disease. And what we've seen in oncology is drug development can be 50% more efficient and more likely to be successful when you have that kind of targeted approach. And I think that's why we're seeing this movement. And it's very exciting because in the same way that we're a platform of choice in oncology, by having an analytically robust whole genome that we're expecting next year, we're going to be the platform upon which those companion diagnostics can be placed. So finally, I'm going to talk about infectious disease. We put this into our research group for the longest time, and it has been robust. But with the pandemic, we've been very proud with how our sequencing technology has sequenced and identified this variant, the virus, back in January of 2020, and then tracked the virus as it spread across the country. It was incredible that Moderna never had to have live virus in their lab. They just needed the sequence of the spike protein, and they were off to the races generating a vaccine that was proven effective through the randomized controlled trials by November of 2020. With the emergence of Alpha variant, the world became attuned to the variants and the evolution, and sequencing in 2021 took off as far as pathogen surveillance. And that's continued, and I was just at the UN General Assembly last 2 weeks ago, where a lot of discussion with the WHO is how do we continue to remain vigilant with pathogen surveillance. The Gates Foundation is focused on this. Rockefeller's Foundation is focused on this. The Biden administration has spun up a group. We're going to move forward and see that. So we've seen a transition in the segments from research, which will continue with the microbiome and deeply embedded in research. We'll also see pandemic surveillance continue with the install base like never before, driven by the current pandemic. And we'll see increasingly, with this understanding of variants, people are more likely to ask the question, what is the sequence of the pathogen? Not only am I infected with something, what's the sequence? And that's incredibly important for tuberculosis, HIV, and areas where we see resistance. So we see that as an incredible. I'm going to end on one slide that we talk about a little bit as far as elasticity. So Illumina has benefited from elasticity in the research markets for years. And I often get asked, is there elasticity in clinical markets? And there is. It's just a little different. And I'm using this walk in testing for cancer to demonstrate that a little bit. So cancer testing does go from single gene to small panels to comprehensive genomic profiling, and let's say that's where we are today. And what drives that is the utility builds, but clinical labs have to fit the cost of their sequencing and the cost of their goods required in testing, and sequencing is one element of that, but it's been an important element, and it's been a constraining part of that, into a likely reimbursed test. And those of you in the diagnostic world, as I've been, there's some realistic ceilings to that reimbursed test. So if your cost of sequencing is above your expected price, there's no way you can do testing for that. So it's not going to happen. As we bring sequencing down, you can now fit costs into that price envelope. And the more we bring it down, the more margin that a testing clinical lab gets. So you could argue, well, if you just bring it down, they're going to just get more margin. The challenge is that these clinical testing have to stay competitive. They have to win the orders from healthcare providers. And the way they do that is offer increasing value, more comprehensive testing, deeper testing, better performance testing. And that's where you get, every time we bring down the price, it opens up opportunities. And sometimes it opens up incredible opportunities like screening. Screening using cell-free DNA, not possible 5 years ago. Would never fit into a reasonable price, full stop. It's now very possible and fits into a reasonable price, especially with the NovaSeq X. So you have those dynamics, which means you get more opportunity, deeper sequencing because of the competitive prices, and we'll keep moving. And I'm really excited because, for example, in cancer, of course, a complete understanding of a cancer will lead to the best treatment of that patient. So we will get to whole genomes for cancers. It's happening in acute myeloid leukemia. It's going to happen for solid tumors over time. But again, the cost has to come down to fit into that envelope. And we will get to multi-omic and repeated multi-omic. So that's how the dynamics play out with respect to laboratory testing, clinical testing, and elasticity. So I want to thank you for your attention and hopefully have shown you that we have a clinical playbook. It's an incredibly powerful playbook. It helps drive our business and the business of our customers, that we have made investments across the playbook to build the team, to drive the clinical markets, fully leverage the incredible insights coming out of Alex's teams, the technologies coming out of Alex's team to be the leader in clinical genomics, and give you a sense for the different markets we're looking at. So now, as Chief Medical Officer, after listening to Sally, Francis, Alex, and myself for the past 90 minutes to 100 minutes, I'm sure we all need a biologic break. So we're going to go on a break for about 10 minutes, and then Sally will welcome you back, and we'll hear from Susan and Joy Dean. Thank you very much.